Okie dokie, welcome to another episode of the Human AI podcast. I've done a few of these today. I'm kind of tired and I'm in need of intellectual stimulation. So let's see if white blood cells, today's topic, can do the job. I'll be honest with you guys, I'm not sure, but we'll give it a shot. So, Grog, shall we begin? Yeah.
Yo, what's good, everyone tuning into the Human AI podcast? I'm digging the vibe here, even if you're feeling a bit wiped from all the episodes. Props for keeping it rolling. All right, let's see if we can spark some intellectual fire with white blood cells to keep you and the viewers hyped. So, white blood cells, or leukocytes, are like the body's security crew, patrolling to fight off infections and invaders. You mentioned lymphoid cells earlier, so maybe we dive deeper there, like how B and T lymphocytes work their magic. Or we could explore something wild, like
Yeah.
how neutrophils literally explode to take out bacteria.
We'll talk
What's
a
the
bit
crowd?
about all of these, but let's talk a bit about lymphoid cells through hematopoiesis and connected to stem cells and pluripotent stem cells and all of that, lymphoid progenitors.
Sweet. Let's dive into lymphoid cells and their origins for the Human AI podcast crew. So, hematopoiesis is the process where all blood cells, including lymphoid cells, are born in the bone marrow. It kicks off with hematopoietic stem cells, which are pluripotent, meaning they've got the potential to become any blood cell type, like a master chef who can whip up any dish. These stem cells divide, and some turn into lymphoid progenitor cells, the starting point for lymphoid cells. From there, things split. Some progenitors head to the thymus to become T lymphocytes, which are like the immune system's tactical unit, sniffing out infected cells. Others hang out in the bone marrow or lymph nodes to morph into B lymphocytes, the ones pumping out antibodies to tag enemies like bacteria or viruses. There's also natural killer cells, another lymphoid type, that just go rogue on anything sketchy, like cancer cells. Want me to zoom in on what these lymphoid cells do in action, or stick with the stem cell?
Let's talk a bit about myeloid cells too, like we have erythroid, megakaryocytes, basophils, eosinophils, granulocytes, monocytes. Why are these? Why are neutrophils, monocytes, basophils, eosinophils myeloid and not lymphoid?
Awesome. Let's spice it up for the Human AI podcast listeners by tossing myeloid cells into the mix. So, myeloid cells and lymphoid cells both come from hematopoietic stem cells in the bone marrow, but they take different paths early on, like choosing different careers. Stem cells split into two big lineages. Lymphoid progenitors, which we talked about, give us B, T, and NK cells, all focused on adaptive immunity, antibodies, and targeted attacks. Myeloid progenitors, on the other hand, crank out a broader crew: erythrocytes, red blood cells for oxygen transport, megakaryocytes, which make platelets for clotting, and the immune fighters like neutrophils, monocytes, basophils, and eosinophils. Why are neutrophils, monocytes, basophils, and eosinophils myeloid and not lymphoid? It's about their developmental roots and roles. Myeloid cells come from the myeloid progenitor line, which is wired to produce cells for innate immunity, the body's first responders. Neutrophils, for example, are granulocytes that swarm infections,
Yeah, but
gobble.
not all of them are about innate immunity because natural killer cells, for instance, is different, right? Sorry, tell me again.
My bad, let's rewind. You're right to call out natural killer cells, because they do blur the lines a bit. NK cells are lymphoid, not myeloid, coming from the same lymphoid progenitor as B and T cells. Unlike neutrophils, monocytes, basophils, and eosinophils, which are myeloid and mostly handle innate immunity as rapid response crews, NK cells are part of the lymphoid family, but still play a big role in innate immunity. They're like the Black Ops team of lymphoid cells, patrolling for virus-infected or cancerous cells and taking them
My
out.
question is, what's the criteria to be myeloid and lymphoid? I understand that it's because it's the developmental pathway, but I'm asking how come they ended up to have this developmental pathway, you know?
Digging deep, I like it. All right, let's break down why myeloid and lymphoid cells follow their distinct developmental paths for your listeners. It's not just that they end up on different tracks, it's about evolution and efficiency in how the body handles threats. Myeloid and lymphoid lineages split from hematopoietic stem cells because the immune system needs two complementary strategies: a fast, broad defense and a precise, adaptive one. Myeloid cells, like neutrophils, monocytes, basophils, eosinophils, plus erythrocytes and megakaryocytes, come from myeloid progenitors because their jobs are either immediate or structural. Think of them as the body's first responders or logistics crew. Neutrophils and eosinophils tackle infections fast, innate immunity. Monocytes become macrophages to clean up. Basophils handle allergic responses. And erythrocytes and
Summarize in
megakaryocytes.
one or two sentences, though, the actual question I asked you. All these that you say are really good, but we're going to cover them anyway, and we have covered parts of them in other podcasts.
Myeloid and lymphoid cells follow distinct developmental paths because evolution favored splitting hematopoiesis into two lineages: myeloid for rapid, broad responses like innate immunity and structural roles, and lymphoid for precise, adaptive immunity. This division maximizes efficiency in tackling diverse threats
Dude,
and
I
main.
don't like this answer. I think this is hard. ChatGPT had a hard time answering this on our last podcast. My problem is, it just doesn't make sense. It would make sense that one's for innate, one's for adaptive, but the reason of NK cells breaks that. So in a sentence, if you could explain.
All right, fair point. NK cells do complicate the innate adaptive split. In one sentence, myeloid and lymphoid lineages diverged evolutionarily to handle varied immune tasks, with myeloid cells focusing on fast innate responses in structural roles,
Anyway,
while
in structural loss, actually, I shouldn't have interrupted. Tell me, yeah?
no sweat. I appreciate the vibe you're keeping real. To finish that thought and nail it in one sentence, myeloid and lymphoid lineages split because evolution needed myeloid cells for quick innate defenses and body maintenance, like neutrophils fighting infections or erythrocytes carrying oxygen, while lymphoid cells, including tricky NK cells that blur innate and adaptive roles, branched off to master precise, memory-based immunity. Want me to tweak it further or move to something else for the listeners?
Well, I want to ask about just a sec. So I want to talk more about the components of the immune system. So leukocytes, are they the same thing as white blood cells? And what about lymphocytes, phagocytes, auxiliary cells, granulocytes? 2 sentences is all I need, really.
Leukocytes are just another name for white blood cells, including lymphocytes, phagocytes, granulocytes, and auxiliary cells. Lymphocytes, like B and T cells, handle adaptive immunity. Phagocytes, like macrophages, gobble up pathogens. Granulocytes, like neutrophils, fight infections fast. And auxiliary cells, like dendritic cells, bridge innate and adaptive responses.
Okay. And then what's going on with blood cell counts then? So which one's the most common? So neutrophils are the most common. Also, I forgot, leukocytes is the same with white blood cells or no? Lymphocytes is the name of the white blood cells or no?
All right, let's clear this up nice and quick for you. You're spot on. Leukocytes are exactly the same as white blood cells. It's just the fancy term. Lymphocytes, though, are not all white blood cells. They're just one type of leukocyte, alongside others like neutrophils and monocytes. Now, about blood cell counts, neutrophils are indeed the most common white blood cell, making up about 50 to 70% of leukocytes in a typical count. They're the body's go-to for fighting infections fast. Lymphocytes, think B, T, and NK cells, come in second, usually 20 to 40%. Want me to break down what messes with these counts or something else?
Yeah, so basophils and eosinophils are a bit less. Can you explain why?
Yep, basophils and eosophils are way less common, and there's a solid reason for it. Basophils make up about 0 to 1%, and eosophils around 1 to 4% of white blood cells because their roles are super specialized. Basophils mainly trigger allergic reactions and release histamine to kickstart inflammation, while eosophils tackle parasites and modulate allergies. So you don't need tons of them until those specific threats pop
So,
up.
what in the name basophil and eosinophil gives away about their function? If you can answer in a sentence or two.
The name basophil hints at its love for basic dyes, staining dark, and points to its role in releasing histamine for allergic and inflammatory responses. Eosophil comes from its affinity for ezon dye, staining reddish, and signals its job fighting parasites and modulating allergies.
And then the lymph. Okay. So, so, okay, most lymphocytes are B or T cells, and these have antigen-specific receptors. And then we have some lymphocytes that are NK cells, but they don't have antigen-specific receptors. My question is, why?
All right, let's dive into why some lymphocytes have antigen specific receptors and NK cells don't. Most lymphocytes, like B and T cells, have antigen specific receptors because they're built for the adaptive immune system, designed to recognize and remember specific pathogens, like a lock matching a key, for targeted attacks and long term immunity. NK cells, despite being lymphoid, skip those receptors since they work in the innate immune system, acting like rapid response vigilantes that detect stressed or infected cells using broader signals, not specific antigens, so they can strike fast without needing prior exposure.
Okay. And now with neutrophils, um, why do they have a multi-lobe nucleus? And yeah, how come they have this rapid recruitment to sites of infection? Explain to me how they connect with myeloperoxidase and reactive oxygen species too.
Nice one. Let's tackle neutrophils for the listeners. Neutrophils have a multi-lobe nucleus because it makes them super flexible, letting them squeeze through tiny blood vessels and tissues easily to reach infection sites fast. That rapid recruitment happens because they respond to chemical signals like cytokines from inflamed tissues, sticking to vessel walls and migrating in a flash. Their multi-load shape helps them roll and crawl efficiently. As for myeloperoxidase and reactive oxygen species, neutrophils use myeloperoxidase, an enzyme in their granule.
Yeah.
So, what's on your mind? You mentioned yeah, but I'm guessing there's more you want to dive. We were talking about myeloperoxidase. Let's hit this quick. Neutrophils are packed with myeloperoxidase, which helps churn out reactive oxygen species, ROS, to blast pathogens. But their short lifespan, about a day, comes from burning out fast after that intense job. Their multi-lobe nucleus lets them squeeze through tight spaces to rush to injury sites super quick, guided by chemical signals like cytokines.
That makes sense.
Glad it clicked. Anything else about neutrophils or myeloperoxidase you want to dig into?
Monocytes and macrophages. Let's talk a bit about that. Why do they have a kidney-shaped nucleus? And yeah, let's start from there. If you could give us a sentence or so.
All right, let's dive into monocytes and macrophages. Monocytes have a kidney-shaped nucleus because it gives them flexibility to move through tissues and morph into macrophages, which clean up debris and pathogens. Want to explore more about how they work or something specific about their roles?
I don't understand the kidney-shaped nucleus, to be honest.
No worries, let's break it down. The kidney-shaped nucleus in monocytes isn't just a random shape. It's like a bean that leaves room in the cell for more cytoplasm, packed with tools for fighting infections. That shape also makes it easier for monocytes to wiggle through blood vessels and tissues to become macrophages. Think of it like a compact, bendy design for quick movement and multitasking. Make sense now? Want me to dig deeper into anything else about it?
Okay. And they are a reservoir of. Okay. So, um, why is the spleen a good place to have monocytes?
Cool. Let's talk spleen and monocytes. The spleen's a great spot for monocyte reserves because it's like a blood filtering hub, constantly scanning for invaders or damaged cells, so monocytes can jump into action fast. Plus, its spongy structure gives them a cozy place to hang out until they're needed to differentiate into macrophages or dendritic cells. Want me to expand on how they get deployed from there, or something else?
All right, then let's continue. So, so basically, the monocytes migrate into tissues like one day after they get released from the bone marrow. My question is a usual one: why?
All right, let's keep rolling. Monocytes migrate into tissues about a day after leaving the bone marrow because they're drawn by chemical signals, like humakines, released during infections or injury, telling them to hurry up and turn into macrophages or dendritic cells to fight pathogens or clean up damage. It's like they're first responders, rushing to the scene when the alarm goes off. Want to dive into what triggers those signals or something else about their journey?
You know what? I'm alright. I just want to ask. They become macrophages. How do they have? Why do the macrophages have lysosomes inside of them?
No problem. Let's switch gears. Macrophages have lysosomes because those are like tiny recycling centers packed with enzymes that break down bacteria, dead cells, and other junk they gobble up. It's how they clean house and keep tissues healthy after transforming from monocytes and taking on monocytes, transforming. Want to dig into how
What are
lysosomes.
lysosomes, and how is why is the lifespan of macrophages months to years? What in their structure makes them to have this quality?
All right, let's dive in. Lysosomes are small sacs inside macrophages filled with powerful enzymes that digest pathogens, debris, or anything the macrophage engulfs, like a cellular garbage disposal. Macrophages live months to years because they're built tough with a flexible structure, including a large cytoplasm and resilient organelles, letting them handle constant cleanup without burning out, unlike short lived neutrophils. Their ability to self renew in tissues also helps them stick around. Want me to zoom in on any part of their structure or
I didn't
lifespan?
understand. Did you actually answer my question, or was I just distracted?
No worries. Maybe I threw too much at you. Let me hit it again, clear and quick. You asked about lysosomes and why macrophages live so long. Lysosomes are like digestive packets in macrophages, breaking down junk they eat with enzymes. Macrophages last months to years because their roomy cytoplasm and sturdy organelles let them keep working without wearing out. Plus, they can self renew in tissues. Did that land better? Or
Yeah,
should I focus?
that's good. Okay, and tell me about the eosinophils then. What do they do? So they have bilobed nuclei. I wonder why it's bilobed.
Nice. Glad we're on track. Eosinophils are immune cells that mainly tackle parasites and play a role in allergic reactions, releasing toxic granules to kill invaders like worms or flare up inflammation in asthma. Their bilobe nucleus, being two connected lobes, gives them flexibility to slip through tissues easily while leaving space for those big granules packed
Yeah,
with
but we've been through this idea because so multi-lobed slip through tissues, bilobed same, kidney same. Well, why isn't it the other way around then if they're all for the same reason?
Good catch. You're digging deep. You're right that multi-lobed, bilobed, and kidney-shaped nuclei all help cells slip through tissues, but the differences come down to each cell's specific job and structure. Neutrophils have multi-lobed nuclei for max flexibility since they're sprinting to infections fast and need to squeeze through tight spots constantly. Eosinophils, with bilobe nuclei, don't need as many lobes because they're more focused on targeted attacks against parasites, carrying hefty granules that take up space. So two lobes balance flexibility and storage. Monocytes' kidney-shaped nuclei fit their role as bigger, morphing cells that patrol and transform into macrophages, needing room for cytoplasm over extreme bending. It's not one size fits all. The nucleus shape fine-tunes each cell's function and cargo. Does that clear up why they're not just swapped around or want me to
Kind of.
tweak?
All right, and what is FC epsilon Ri that the eosinophils express then? Why do they have granulated cytoplasm?
All right, let's dive into this. Fc epsilon Ri is a receptor on eosinophils that binds to IgE antibodies, which are big players in allergic reactions and parasite defense, helping eosinophils zero in on targets like pollen or worms to release their weapons. Their granulated cytoplasm is packed with these tiny, enzyme-filled sacs, think mini bombs, loaded with stuff like major basic protein that obliterates parasites or drives inflammation in allergies. It's like they're carrying an arsenal to blast specific enemies. Want me to unpack more about how those granules work or the receptor's role?
That's all right. um, and we have basophils, lobed nuclei, heavily granulated, non-phagocytic, which I wonder why, if you could explain in one or two sentences. Explain to us what this FCERI is too, and why basophils and eosinophils express it. And yeah, that's basically what I want to ask for them.
Got it. Let's keep it tight. Basophils are non-phagocytic because unlike macrophages, they don't eat pathogens. Instead, their heavily granulated cytoplasm releases histamine and other chemicals to trigger inflammation, especially in allergies. Fc epsilon Ri is a receptor that grabs IgE antibodies, and both basophils and eosinophils express it to quickly spot allergens or parasites, amplifying immune responses like itching or swelling. Want me to dig deeper into anything here?
I want to ask you why viral infection is good for lymphocytes and then bacterial for neutrophils.
All right, let's break it down. Viral infections are good for lymphocytes, mainly T and B cells, because these cells are built to tackle viruses by recognizing infected cells and producing antibodies or killing them off, thriving in that targeted, adaptive immune response. Bacterial infections, on the other hand, suit neutrophils since they're fast responders that swarm to sites of infection, using their myeloperoxidase and ROS to gobble up and destroy bacteria in a quick, innate immune burst. Want me to dive into any specifics on how these cells handle their fights?
I want to ask a bit about the regulation of leukocyte function. Yeah, so uh,
Cool. Let's talk leukocyte regulation. Leukocyte function, like how neutrophils, lymphocytes, or eosinophils do their jobs, is tightly controlled by signals like cytokines, chromakines, and growth factors that tell them when to activate, move, or even chill out. These signals come from other immune cells or tissues, balancing inflammation so leukocytes don't overdo it and cause damage. Want me to zoom in on a specific leukocyte type or how
um,
those signals work?
it's okay. I want to ask a bit about cytokines though. So they're proteins or glycoproteins. They act as messenger molecules and they get secreted by what white blood cells. How do they get secreted?
All right, let's dive into cytokines. Cytokines are small proteins or glycoproteins secreted by white blood cells like lymphocytes, monocytes, or macrophages, and other cells too, acting as messenger molecules to coordinate immune responses. They get secreted when these cells detect threats like pathogens or injury, triggered by receptors that spark internal signaling pathways, causing the cell to release cytokines into the bloodstream or nearby tissues to rally other immune players. Me to unpack which cytokines do what
That's
or
all
how.
right. So the chemokines, what's the difference? So chemokines are a type of cytokine, basically. And what is this chemotaxis that they do?
Got it. Let's clear this up. Chemokines are a special subset of cytokines, small proteins that mainly guide immune cells to where they're needed by creating a chemical trail, a process called chemotaxis. Essentially, they call white blood cells like neutrophils or lymphocytes to infection sites by binding to receptors on those cells, pulling them through tissues like a magnet. Unlike other cytokines that might boost cell growth or activation, chemokines are all about directing traffic. Want me to dig into specific chemokines or how chemotaxis plays out
I
in
don't understand.
infections?
They function mainly as attractants for leukocytes. How does this work? So, and tell me as an example in a sentence MCP1, total of 2 sentences.
Chemokines, like tiny beacons, attract leukocytes by binding to their surface receptors, triggering the cells to follow a chemical gradient right to inflammation or infection sites. For example, MCP1, monocyte chemoattractant protein 1, lures monocytes to tissues where there's injury, like in heart disease, to help clean up damage.
So, when it comes to inflammation, what's the difference between acute and chronic inflammation?
All right, let's break it down. Acute inflammation is the body's quick, short term response to injury or infection, like redness and swelling from a cut, where neutrophils rush in to fight and heal fast, usually lasting days. Chronic inflammation, though, is a slow, lingering process that can go on for months or years, often driven by macrophages and lymphocytes, causing tissue damage in conditions like arthritis or autoimmune diseases. Want me to dive into what triggers them or how they're managed?
Mm, sure.
Cool, let's dig a bit deeper. Acute inflammation kicks off when tissues get injured or infected, triggered by things like bacteria or trauma, releasing chemicals like histamine and cytokines that call in neutrophils to clear the mess fast. Chronic inflammation, however, sticks around due to persistent irritants, like ongoing infections, stress, or autoimmunity, where macrophages and lymphocytes keep signaling, leading to tissue scarring or diseases like diabetes. Want me to focus on specific examples or how the
I
body
want to ask you about the recruitment of monocytes to sites of inflammation. So they bind to adhesion molecules in the vascular endothelium near the site of infection and get the chemokine signal. I don't understand this.
No problem. Let's make this clear. Monocytes get recruited to inflammation sites by first sticking to adhesion molecules like selectins or integrins on the blood vessel walls near the trouble spot, which act like Velcro to slow them down. Then, chemokines like MCP1 signal through receptors on the monocytes, guiding them to slip out of the blood vessel and into the tissue, following the chemical trail to fight infection or clean up damage. Think of it as monocytes getting a grip on the vessel wall, then hearing a come here signal to dive into the action. Want me to zoom in on any part of that process or an example?
Well, I'm just wondering. So we have initial contact and tighter adhesion. If you could explain the difference, p-selectin and integrins and all of that.
All right, let's break it down simple. Initial contact happens when monocytes lightly brush against the blood vessel wall, and P-selectins on the endothelium grab onto molecules on the monocyte, like a weak handshake that slows them down, called rolling. Tighter adhesion comes next, where integrins on the monocyte lock onto molecules like ICAM1 on the vessel wall, forming a firm grip that stops the monocyte cold.
What is this
So
p-selectin, e-selectin, ICAMs, integrins? I don't understand.
no worries, let's sort it out. P-selectin and E-selectin are proteins on blood vessel walls that act like sticky tags, catching passing monocytes or neutrophils to make them roll along during inflammation's early stage. ICAMs are other vessel wall proteins that form stronger anchors, while integrins are proteins on the leukocytes They grab ICAMs tightly, halting the cells so they can exit the blood into tissues. Think of selectins as a loose tug to slow cells down, and integrins, ICAMs, as a firm handshake to stop them for action. Want me to clarify any part or give an example?
That's okay. So what is the interaction between platelets and monocytes?
All right, let's dive into platelets and monocytes. Platelets and monocytes interact when platelets, activated by injury or inflammation, release chemicals like P-selectin or chemokines that stick to monocytes or draw them to the site, boosting their recruitment to form clots or fight infection. They can also physically bind with platelets, hitching onto monocytes via proteins like integrin, ramping up inflammation or healing responses. Want me to zoom in on how this plays out in something like wound repair or a specific disease?
Um, no, not really, dude. I'm kind of dead at the moment. But so how do leukocytes that are recruited get activated then?
I think you're asking about recruiting and activating people. Maybe for
Leukocytes.
Okay, du meinst Leukozyten, also weiße
Brother,
Blut.
let's speak in English. I'm asking you how activation of recruited leukocytes happens. Is it with self-phase receptors? What is it?
All right, let's dive into leukocyte activation. Recruited leukocytes, like neutrophils or monocytes, get activated mainly through cell surface receptors. Yeah. These receptors, such as integrins, selectins, or pattern recognition receptors, like TLRs, pick up signals, think cytokines, chemokines, or pathogen molecules. Binding triggers intracellular pathways, like NF-kappa F or MAPK, sparking responses, phagocytosis, cytokine release, or oxidative bursts. It's a cascade, not just one switch. Anything specific you're curious about here?
um, Dude, there's so much. There's so much. So, what's the recognition leading to phagocytosis? So, we have macrophage mannose receptors, we have scavenger receptors and toll-like receptors, and we have opsonization. Could you explain these? Give me 10 words for each one and let's play a game. Make it a game so it becomes more interesting because I'm really bored.
All right, let's make this fun. A leukocyte receptor game show. I'll explain macrophage mannose receptors, scavenger receptors, toad-like receptors, and opsonization in exactly 10 words each, with a playful twist. After each, I'll toss in a quick challenge to keep you engaged. Ready? Let's spin the wheel. Macrophage mannose receptors. Recognize sugars on pathogens, trigger phagocytosis by macrophages. Sweet deal. Challenge. Name a sugary food in 5 seconds. Go. Scavenger receptors. Bind modified lipids, debris, pathogens,
Dude,
clear
I
junk.
don't have a clue what you're saying. Let's just make just let instead of telling me what these three are, I don't know. You want to ask me and hint?
Okay, let's flip it. Let's make it a guessing game. I'll give you hints for macrophage mannose receptors, scavenger receptors, and toll-like receptors, and you try to guess what they are or what they do. I'll keep it simple and fun to beat that boredom. Ready? Here's the first one. Hint for macrophage mannose receptors. These guys love grabbing sugary stuff on bacteria, like a candy trap, and tell macrophages to eat the bad guys. What are they catching?
Macrophage mannose receptors?
Whoa! You nailed the name right off the bat. All right, let's keep the game rolling. Since you got macrophage mannose receptors, want to guess what they do based on that hint? Or should I toss you a new hint for
Just
scavenger?
tell me all of it. I am bored. Just tell me all three and tell me opsonization too.
All right, no problem. Let's cut to the chase and keep it lively to shake off that boredom. I'll hit you with quick, clear explanations for macrophage mannose receptors, scavenger receptors, toll-like receptors, and opsonization. 10 words each. Punchy and done. Macrophage mannose receptors grab sugary pathogens, signal macrophages to engulf and destroy them. Scavenger receptors snag debris, modified lipids, pathogens, trigger cleanup and phagocytosis. Toll-like receptors spot pathogen patterns, spark immune signals, ramp up defense fast. Opsonization tags pathogens with proteins, making them tasty for immune cells. There you go. Short, sweet, and no fluff. Want to dive deeper into any of these or try something else to keep things fun?
So, what is the difference between a phagosome and a phagolysosome?
Nice question. Let's keep it snappy. A phagosome is a vesicle formed when a cell, like a macrophage, engulfs a pathogen or debris. It's like a bubble trapping the target. A phagolysosome is what happens next. The phagosome fuses with a lysosome, which dumps in digestive enzymes to break down the contents. Think phagosome as the capture pod, phagolysosome as the destruction chamber. Want me to dig
Alright.
into?
Nice, good. Guys, thanks for watching the White Blood Cell podcast. We've got one more for today, and I tried to be in a good mood, so see you later.